Plasmid PUC19. Plasmid confer resistant to ampicillin and can make functional beta galactosidase restriction cut in the polylinker DNA fragment. Plasmid confer ampicillin resistant but can't make functional beta galactosidase. This is DNA ligase, DNA insertion in the disrupted lect Z gene. Phage lambda cloning vector, engineering version of bacteriophage lambda infect E. coli. Central lesion of the lambda chromosome linear is cut with restriction enzyme and digested DNA is inserted. DNA is packaged in phage head to form a virus particle. Phages with both ends of the lambda chromosome and 37 to 52 kilobyte insert replicate by infecting E. coli. Phage replicate using E. coli and the lytic cycle. This is the cycle. Like plasmid vector, large number of restriction site available, useful for large DNA fragment than PUC19. Release of the progeny, infection, phage DNA, lytic response, lambda chromosome replication, breakdown of those DNA, replicate lambda chromosomes, lysogenic response, integration of lambda chromosome into the host chromosome, multiple division, induction of lytic cycle, excision of lambda chromosome, and enter the lytic cycle, breakdown of the host DNA. This lambda chromosome replicate. This is the life cycle. The end is assembly of the progeny of lambda chromosome, lambda phage. Here we treat it with echo R1. Arms contain all the genes needed for replication but are too small for packaging of DNA in lambda particles using protein and enzyme from the lambda infected E. coli. Cosmic cloning vector, feature of both plasmid and phage cloning vector don't occur naturally. Circular origin, here the origin, selectable marker, ampicillin resistant gene, cloning sites, cause site, permit packaging into lambda phage and introduced to E. coli cells, subtle vectors capable of replicating in two or more types of host, replicate autonomously or integrate into the host genome and replicate when the host replicate. Commonly used for transporting gene from one organism to another, as forming animal and plant. Insert carefully luciferase gene into plasmid and transform agrobacterium, grow agrobacterium in large quantities and infect tobacco plant. Yeast artificial chromosome, vector that enable artificial chromosome to be created and cloned into yeast. Feature, yeast tolerate at each end, yeast centromere sequence, selectable marker, amino acid dependence in each arm, autonomously replicating sequence. For replication, restriction site for DNA ligation, useful for cloning very large DNA fragment up to 500 kilobase per, useful for very large DNA fragment. Bacterial artificial chromosome vector that enable artificial chromosome to be created and cloned into E. coli. features useful for cloning up to 200 kilobase pair, but can be a handful like regular bacterial plasmid vector, useful for sequencing large stretch of chromosomal DNA. Like other vectors, bacterial artificial chromosome contain erogen sequence derived from an E. coli plasmid called F factor, multiple cloning site, restriction site, and selectable marker antibiotic resistance.